Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Clinical and Bioanalytical Aspects of Immune Monitoring via ELISpot. My name is Sydney, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Celerion, who developed the content for this presentation. Celerion, a global leader in early clinical research services, offers a unique combination of medical expertise, clinical operations experience, and scientific excellence that gives its clients the confidence to make fast, accurate decisions about their drug development path. For 50 years, Celerion has leveraged the latest operational concepts and technologies to execute safety and tolerability, pharmacokinetic and pharmacodynamic studies in highly controlled clinic environments. Celerion offers feasibility, data management, biostatistics, clinical monitoring, and bioanalytical services. Its founding mission is to help its clients get their drugs to market quickly so that they touch the lives of our family, friends, and people around the world in need. And now I'd like to introduce our speakers for today's event. Wendy Adamovich is Senior Scientist in the Molecular and Cell Biology Group within Bioanalytical Services at Celerion. Her focus is on method development and validation of ELISpot, qPCR, flow cytometry, and biomarker assays. Wendy joined Celerion in 2016, bringing over 20 years of pharmaceutical experience in assay development and target validation for both large and small molecule programs in oncology, psychotherapeutics, and neurodegeneration. Arnout van Harst has over 25 years of drug development experience. As Director of Scientific Affairs, he plays a key role in supporting clients to design drug development studies through proof of concept. As a member of Celerion's research team, in particular respiratory and vaccines, he works closely with external experts in the fields of pulmonology and vaccine development. And now, without further ado, I'd like to hand the presentation over to our first speaker. Arnaud, you may begin when ready.
Thank you, Sydney, and welcome to this webinar about immune monitoring via ELISpot, during which we will touch upon clinical and bioanalytical aspects. Now, I will introduce you to Celerion and also take you through some of the clinical aspects around immune monitoring via ELISpot. And then, as said, my colleague Wendy Adamovich will take over, after which we will have some more time for questions and answers before concluding this webinar. Celerion is one of the leading global providers of early clinical development services, with key expertise in therapeutic areas like vaccines and respiratory drug development. And for the conduct of and management of clinical trials, including multi-center trials, we combine our own in-house clinical trial units, clinical pharmacology units, together with external qualified sites that often are at academic sites. We're a full service organization, meaning that we can support basically all the services around your clinical trial, including study design and data management services, data reporting and report writing, things like that. As Wendy will allude to later, we also have a strong bioanalytical group, but I will first show this map, which illustrates how we have a global presence, with particular attention to the the clinical sites, our own in-house phase 1 units with an overall capacity of around 600 beds, which are based in the US, in Lincoln and Phoenix, but we also have a clinical research facility in Belfast in the UK. And in addition to that, I want to highlight the 2 bioanalytical labs that we have, one in Lincoln and the other in Zurich, Switzerland. We're a well-recognized partner to biotech, innovative biotech and pharmaceutical industry, and we provide comprehensive data early in the process of clinical drug development, enabling our clients to take early go, no-go decisions for their new products. This graph on the right shows that the majority of our clients are biotechs, which is probably also reflective of a good cultural match because we're a very lean organization. I mentioned exploratory clinical trials as our main field of interest, and this would encompass particularly healthy volunteer studies, but also patient studies. And typically we have great experience in running first-in-human studies with both small molecules, but also biologics. And we're familiar with all kinds of complex study designs, such as hybrid studies, where we conduct single and multiple ascending dose studies in normal healthy volunteers, and occasionally also in patient panels. In addition, we do lots of proof of principle studies, which often, as will be part of this story, will encompass biomarker assessments, and we're very familiar to umbrella protocols, so that are studies that are integrating a multiplicity of factors, such as internal and external factors, like drug-drug interactions, the effect of smoking or of food, but also evaluations in special populations such as obese and elderly. This is just a figure to show how exploratory drug development is seen as in the stage of development. Like on the top, you can see the traditional paradigm of phase 1, 2, and 3 up to drug registration. And the exploratory phase is really where we are focusing, which basically addresses all the research around a new drug to assess the characteristics and the early effects of the drug, exploring those before proceeding to the confirmatory late-stage trials. And in these studies, although safety and tolerability are the typical main endpoints in the initial development, many of these studies will try to establish additional proofs. So, for instance, proof of presence. So, does the drug under study also reach its site of action? Or proof of mechanism. So, in addition to pharmacokinetics, does the drug also affect its biological target as the molecule was designed? And at a higher level, one can even look at proof of concept studies, where one wants to know whether a drug also favorably impacts the disease or has also an acceptable risk of toxicity. And from top to bottom, these types of proofs will increasingly be established in a target population or a patient population rather than only healthy volunteers, and also will include evaluation of biomarkers, either to reflect the impact on disease or to reflect toxicity. Now, this is also where ELISpot comes into play, which can play a great value or add great value in terms of functional responses to the immune system, or to monitor the antigenicity of biological therapies, for instance. Now, ELISpot is a very flexible and functional assay to evaluate the functional status of individual cells. It was originally developed to detect antigen-specific antibody secretion by B cells or secreting B cells, but is nowadays often applied to evaluate T cell function, so to quantify the cytokine secretion by or the number of T cells in blood samples. responding to a specific antigen. So, for instance, cell cytotoxicity mechanisms may, for instance, involve the release of perforin or granzyme B that are released by CD8+ cytotoxic lymphocytes or natural killer cells, which may induce the killing of a virus-infected cell or a tumor cell. One can also look at T cells secreting certain cytokines, such as a series of interleukins or TNF-alpha or GM-CSF. But most commonly may be applied is the secretion of interferon-gamma as a response to specific antigens by T cells. Now, because it's a very sensitive method, one can also detect low frequency of events in mixed populations of peripheral blood mononuclear cells, so PBMCs. And therefore, application is in a broad set of clinical development fields. Just briefly introducing you to the ELISpot methodology, and I think Wendy will go into more detail there. But basically, this requires collection of blood samples from a healthy subject or a patient, from which the PBMCs are isolated. Then separately antibodies specific for an antigen are fixed or are immobilized to a solid surface, such as a 96 well plate. And then the cell suspension from the blood sample is added to the plate, and a stimulant is also added if appropriate, following which the plates are being incubated. During which the cells will release the actual analyte of interest, so for instance interferon-gamma, which will then bind or be trapped by the antibody on the plate. And subsequently, the cell suspension will be washed away, and a second antibody, often a bioattenuated antibody, is added, which will also recognize the analyte. And then one can proceed to signal amplification, for instance, with avidin. And the subsequent conversion of a chromogenic substrate is what actually generates a colored spot, as you can see on the right side of this slide, where every single spot corresponds with a single cell. Now, in a 2007 publication from Lee and co-authors, ELISpot was, for instance, used in detecting or in demonstrating autoimmunity of patients with emphysema. So they developed autoimmunity against elastin, which is which normally has a role in maintaining the integrity of lung tissue. And they isolated peripheral CD4 plus T cells from the patients and determined cytokine responses to a series of stimulators, including elastin. And in their paper, they describe how, in response to elastin, but interestingly not collagen or albumin, the T cells from the emphysema patients released interferon-gamma. And also there was an association with antibodies in the blood that recognized elastin. And finally, they also demonstrated that the responses in T cells correlated with the actual severity of the disease, of the lung disease, as assessed by CT scans. So in clinical trials, ELISpot may add value by confirming the drug's mechanism of action or provide supportive evidence to the drug efficacy or an immune correlate of protection in case of vaccines. One can also, for instance, as mentioned earlier, monitor immunogenicity of new therapies as a means of undesired effects. So on the left panel, one can see that over the past 20 years, there is a steady increase of the use of ELISpot as an endpoint in clinical trials. And the right panel shows that these trials were particularly exploratory in nature. So from phase one up to phase 2, as shown by the green, black, and grayish areas on the right. Now, if we zoom into the interventional studies and look at what therapeutic areas they were covering, then maybe not surprisingly, ELISpot was particularly applied in fields where with therapeutic intervention that interact with the immune system, such as infectious disease or immunology, and particularly also oncology. But when, because the involvement of the immune system or immunological pathways is relevant in many diseases, there's probably loads of other studies or therapeutic areas that can be relevant or of interest, for which ELISpot can be of interest in its assessment. Now, I alluded to B-cell ELISpot initially. This can, for instance, be used in vaccine research by determining the peak response in antibody following immunization. But it can also help identify the optimal antigen or the best antigen or adjuvant that can trigger a B-cell response. Memory B cells can be assessed to determine whether a lasting response can be established, and one can also, ELISpot can also help differentiate the types of immunoglobulins that are being released, so IgM or IgG, for instance. Interferon-gamma ELISpot assays have been extensively used to study immune responses to vaccines. So for instance, for preventative diseases such as preventative vaccines for diseases such as malaria and tuberculosis. But more recently, in the past 2 years, it has also intensively been used in COVID-19, so in the development of vaccines against the SARS-CoV-2 virus. Now, in the clinic, and the graph shows how the temporal responses, both antibody and T-cell immune responses, may look over time, but these can be very heterogeneous in individual patients or among patients. So ELISpot can add value here by assessing cellular immunity in addition to simply antibodies. And usually antibodies are a sign of past infection in people who have been exposed to the SARS-CoV virus. But T-cell responses are important also to monitor because they are critical in controlling disease in natural infection. And moreover, the illness severity has been shown to correlate with the T-cell responses against for anti-M or anti-S viral proteins. And in vaccine development for SARS-CoV-2, for instance, ELISpot has been applied in many for many vaccines. So, for instance, in at least 10 SARS-CoV trials with different vaccines, ELISpot has been used, showing the release of the response in interferon-gamma, and as shown in this slide. On the left, for instance, for the Sputnik vaccine, and on the right for the AstraZeneca Oxford vaccine, there were robust spike protein responses for T cells as determined by ELISpot. In addition to methodologies like these, one can also apply intracellular cytokine staining with flow cytometry to help identify specific cytokine-producing cell populations. Another field where ELISpot can add value is in gene therapy. Gene therapy is, for instance, based on adeno-associated viruses, where the vector contains viral capsid proteins that can elicit a cellular immune response. But there can also be pre-existing immunity against the wild type of the virus that may also interfere with the therapy. Now, in a trial evaluating the effect of a gene therapy coding for blood clotting factor 8, a specific cellular immune response was detected against the vector capsid proteins. But for this study, a specific interferon-gamma ELISpot was developed and also validated. And interestingly, the study showed that in 15 out of 16 patients that were treated with this gene therapy, a positive response was developed in that these patients showed positive T-cell activity already after 2 weeks after the treatment. But also the majority, the response was only temporarily, and the majority of patients reverted to being negative again after 52 weeks or after a year. The transgene product, which may also elicit potentially an immune response, was much less an issue. Only 5 out of 16 patients showed an intermittent response. This then is an example of how ELISpot can also be very useful in development of cancer vaccines, or maybe more general in immunotherapy trials. This is an example of a trial that was actually evaluating the potential of interferon alpha 2b and GM-CSF as cytokines that were thought that might augment the actual immunological response to a peptide vaccine to be developed for metastatic melanoma cancer, melanoma. And for this study, they used ELISpot as the primary endpoint to evaluate the immune response. Now, the study showed that although these two cytokines did not enhance the immunomodulatory effect of the vaccine itself, interestingly, the immune response to vaccination did correlate to the overall survival, which was prolonged in patients who developed a positive immune response that was assessed by ELISpot in this assay. So this illustrates how valuable the application of ELISpot can be in cancer vaccine trials as well. Now, with that, I want to conclude. So ELISpot is a very sensitive measure of single cell responses. It can help quantify the antigen-specific response. And therefore, it's a valuable endpoint in particularly exploratory clinical trials. But because of the broadness of immune response involvement in many therapeutic areas, ELISpot can be of relevance to a broad set of therapeutic areas. And with that, I want to conclude, and I want to hand over to Wendy. Thank you.
Thank you, Arnott. Today, I'm going to be discussing the bioanalytical aspects of ELISpot. First, I'd like to give you a brief overview of the bioanalytical group where I work. We have the traditional small molecule capabilities, as well as ligand binding, and additionally MCB group, which is molecular and cell biology, where we cover qPCR, cell-based assays, flow cytometry, as well as ELISpot. To give you an overview of what I'll be covering today, I'll start with guidance for fit-for-purpose validations and the ongoing efforts in the field, sample analysis, challenges and solutions that we've encountered as we work through ELISpot projects, and finish up with key takeaways. Arnott gave a great introduction to the ELISpot workflow. I'd just like to highlight a few aspects. In panel A, you can see is the isolation of PBMCs. One critical component of the isolation of PBMCs is the time from collection of blood to the preparation and centrifugation on the density gradient. If you notice in the bottom panel, after centrifugation, the red blood cells and the granulocytes collect at the bottom of the tube, and the PBMC layer is located just under the plasma layer. If you do the centrifugation within 8 hours of collection, This ensures that the granulocytes locate to the bottom of the tube, but after 8 hours, significant numbers of granulocytes become activated, and then they become more buoyant and will co-localize with the PBMC layer, and that can cause a problem. The granulocytes can inhibit T cell function, as well as they're diluting out the PBMC so that your spot number will not be accurate. Arnott gave a great introduction and explanation of panel B, which is the assay. Oh, I forgot to say that traditionally PBMCs are cryopreserved. Once they're isolated, they're cryopreserved, and that way they can be shipped if ELISpot is being carried out at a separate lab. And this is important for multi-center trials. Moving on to the ELISpot assay itself, I'm not going to cover in super detail, as Arnott went into really nice explanation of coding, and then we mix the PBMCs with our treatment. We generally, for interferon-gamma, incubate the plates overnight in a CO2 incubator, and then the cells are washed away, and it's developed pretty much like a traditional ELISA. So that in the right-hand panel, where you see a spot, that's really the collection of interferon-gamma from an individual cell that it secreted overnight in the incubation step. And then finally, we move on to counting and scanning and counting of the plate. The software is quite complex for ELISpot, as you can see from the example here. It's important during method development and qualification steps to optimize scanning parameters, optimize your counting parameters, and this can be done using the software highlighting which wells are the negative control wells and which are the positive, and that allows setting of the different aspects of the counting of the spots. And then finally, the software can also accommodate edge effect compensation, as well as fiber removal or cracking of the membrane. So there is no FDA guidance currently for ELISpot, but at this time, the Global CRO Council and WRIB are working on white papers. There are quite a few guideline papers that have been published over the years. I particularly want to highlight on the right-hand side, the paper by Mecker, and they did some very careful analysis looking at precision and linearity, and I'll show you examples of how we carried that out. Just looking at current guidance for LBA, the BMB guidelines from 2018, you can see a list of components for method validation on the left. Some of these are not applicable to ELISpot, others can be modified to fit our specific needs. So we don't have a reference standard, and we do not have a calibration curve, so those are not applicable. We are able to calculate accuracy, and it is relative accuracy, and we do this by using proficiency testing. Our sensitivity we do by calculating a limit of detection, and then we follow with statistical testing compared to the negative control. For specificity, we can look at both positive and negative peptides to show specific response, as well as it is important to test peptide pools for your antigen of interest in normal and potentially disease populations, but those can be challenging to find. I'd like to go on to the next slide where I have some definition. So we do use a relative accuracy using proficiency testing or inter-lab testing. For precision, I'd like you to note the figure from the Mecker paper on the left side of the screen. So we do measure precision in the traditional way, looking at intra and inter-assay precision, as well as inter-operator. For this, it's important to use the exact same lot of PBMCs and your quality control peptides. And the CV, as shown in the figure here, is really only useful above 30 spots. And that's due to the mathematical certainty that as you're dividing by a smaller number, it makes your CV balloon out. And I'll show you a good example of that later in my data. For specificity, we did use a negative control peptide, and we chose human skeletal actin, which can just be purchased commercially. And in the case of AAV or your vaccine of interest, we also can test peptide pools, and this can be tested in a normal population to see what the background reactivity might be, or relevant or diseased population if available. The LOD is calculated as 3 times the medium spot count for the media-only wells, and this is across the entire validation. And then once you get to sample testing, statistical analysis will confirm your response by comparing back to the media only. Finally, the ULOQ is the maximum spot number that can be differentiated in the software. And then linearity is where you see a proportionality of signal. For example, if you have 100 spots at 100,000 cells per well, you'd expect 200 spots at 200,000 cells per well. And I'll show you the data for that as well coming up. So combining the applicable BMV guidance as well as the various publications in the field, we developed target criteria. First, for precision. For samples that are above 30 spots, we'll use both the standard deviation and %CV. And for wells fewer than 30 spots, we'll just report the standard deviation. And then for spot count of 30 to 100 spots, the CV will be less than 50%. And for a mean spot count of greater than 100, the CV will be less than 25%. For specificity, the expected outcome of the negative control peptide and the medium background control is to have little or no reactivity of less than 10 spots, although occasionally you do see donors that will go above that 10 spot mark. For LOD, as I said before, we use 3 times the median of the background across all the validation, and statistical testing does not occur below the LOD. And linearity is defined as the cell number where we see proportionality, and the results are linear. To give you an idea of the methods we used, PBMCs were purchased from a commercial supplier. Our positive controls were CEF, which stands for cytomegalovirus, Epstein-Barr, and flu peptide pool, as well as a peptide pool corresponding to the PP65 protein of CMV. And finally, PHA is a chemical mitogen and causes a general increase in T-cell reactivity, which is a good control for each plate. Our negative controls are medium, which is the background of the assay, and then we also tested human skeletal actin peptide pool. We incubated between 18 and 24 hours, and 200,000 cells per well were used, other than the linearity assessment where we varied the cell number. And finally, we used pre-coated plates, and this just allows us to save a day and time in the assay. Here's the inter-assay precision data for validation. We ran 4 batches looking at 3 different donors. This was carried out over 2 different days and with 2 different operators. If you look at the bottom row of data here, you can see the reactivity to both CEF peptide as well as the PP65 peptide pool. And I've highlighted in bold where the spot number was greater than 30. And you can see we calculated the standard deviation and %CV. I thought this was also a good example of the description of how the CV can balloon as you get closer to lower spot numbers below 30. You can see for the 1, you have a 90% CV, and under donor number 3, the 2.4 also has a very large CV, and then as you're moving closer to 30 at 15 spots, the CV is starting to come down. The linearity data is shown here. If you look at the bottom graph at the bottom, we tested from 25,000 cells up to 400,000 cells per well. In the next column, you can see the spots per well decreasing as the cell number decreases. And then to look at proportionality, the numbers were normalized to 1 million cells. So if you look along the bottom row at 25,000 cells per well, you can see the proportionality is really dropping off there. And so in the graph at the top of the slide, we show the linear response between 50,000 and 400,000 cells per well, and we were able to calculate an R-squared for that. This shows the specificity. We used the skeletal actin peptide as a negative control peptide to look at the background reactivity and test specificity. The cells for 3 different donors were tested with a CEF peptide pool, PP65, the skeletal actin, as well as the media negative control. You can see we had a good response with CEF and then a background level response with actin and the media control. And these particular donors didn't have a strong reaction to PP65. So to summarize our validation, we were able to develop a validation plan using target criteria based on the BMV, as well as peer-reviewed articles and harmonized guidance from the field. Our precision met the criteria with less than 25% CV for donors with a spot count of over 100 spots per well, and we had an interbatch range from 10.4 to 13.9% CV. We demonstrated specificity using the negative control actin peptide pool, as well as the media only control, and we did have less than 10 spots per well for those. Our linear range we defined as 50,000 to 400,000 cells per well, and this can be important if you have a very low yield of cells, or particularly in pediatric samples, you might have a very small sample size, so it's important to define where you see linearity and proportionality for various cell numbers. And finally, the LOD of the assay was determined to be 12 spots per well, and below this we will not statistically test. The LOD can be different in study samples, particularly if you're using a disease population, so it is recommended that you calculate an in-study LOD. Moving on from validation to sample analysis, I'd like to cover a few important points. First is the quality of the PBMCs. It's really important to have consistent procedures, especially if you have multi-center studies. You want everyone to be following the same, following closely to the same procedure so that you do have high quality PBMCs for the ELISpot assay. And again, just to highlight, you really want to have processing carried out within 8 hours due to the granulocyte contamination risk. If that's not possible, there are tubes on the market called CPT tubes. These contain a frit, and they are processed within 2 hours at the collection site and spun, and then they can be shipped overnight at room temperature. At Celerion, we use a document called the BSP, or Bioanalytical Study Plan, and this is agreed upon with sponsors prior to the start of sample analysis, and it describes a variety of components of the sample analysis. Minimum viability of the sample that will be used, we use greater than 70% based on the literature. And also, it's important to have a priority of plating if your yield of cells is too low. It also describes the in-study LOD and the statistical analysis to be used once the data is collected. Also, the plate design, which should include the pre-dose sample on every plate, and you really want to group the samples by subject, so you always have a nice comparison between pre-dose and the time points of the samples if they're collected through time. And finally, we chose to use the distribution-free resampling, or DFR test. This is an ELISpot specific test from harmonized guidance in the field, but I have seen many papers where they use other types, so this is something, a good discussion between the sponsor and the CRO. Finally, we include a reference sample for each day that ELISpot is run. This is a control lot of PBMCs. The way we approach it is to purchase a large quantity of one single lot of PBMCs, and then they're tested with the same positive control peptide, and that can provide trending data through validation and sample analysis with that large volume of lots of the PBMC. And finally, this trending data is expected by regulatory bodies. I've seen some labs that run once a month, but we run it every single day that we run ELISpot. It can be really helpful if looks like there's a problem with a certain sample. If you have that reference sample, it really helps to troubleshoot. So I'm going to give you an example of the reference sample and data. This shows 8 runs that we carried out over several months. And you can see on the right, there are mean spots per well and the intra% CV for each of the triplicates that was run for the ELISpot. And then at the on the lower right, you can see the results of 73 spots per well is the mean, and the percent CV is 26%. And this does fall within the criteria we set of a percent CV less than 50% between 30 and 100 spots. On the bottom part of the slide, you can see an example of a plate setup, how we run ELISpot here at Celerion. For this, we use example subject 1. We have a pre-dose sample, followed by day 8, and then day 28, and then we include the reference sample at the end of the plate. The various treatments are medium, which is our negative control, CF peptide, our PHA positive control, as well as pools. And these pools can be peptide pools corresponding to your antigen of interest, for example, for a vaccine trial, or it could be from an AAV if you're doing a gene therapy trial. So some of the challenges we've come across and mitigations as we've run ELISpot. First is to have really clear communication with the sponsor of how ELISpot will fit into the clinical study protocol. As I said, the time to processing is quite important. You want to make sure you have an adequate sample size so that you are able to run that pre-dose sample for all the time points. Assessment throughout the course of a trial, which can run up to 1 to 2 years in some cases. It's important to have the correct sample tubes. If you're going to ship, you might want to use CPT tubes to avoid that granulocyte contamination problem. And ensuring that all these happen will give you a quality result in the end. Second is to have trained staff. ELISpot is a complex assay, as well as the software, which is also complex. And it's really important to have sufficient staff, time to train them, to really get a consistent workflow, which is important for efficient work through validation and sample analysis. And finally, for compliance and documentation, most electronic notebooks were designed for either LBA or LCMS work, and they don't always work very well for ELISpot. So we were able to work with our internal resources and design ELISpot-specific electronic notebook forms, as well as validated Excel spreadsheets, so we could have efficient workflow and data processing. So just to summarize key takeaways for the bioanalysis portion of our talk today. First, we designed a fit-for-purpose validation, and you want this to be appropriate for both validation and sample analysis, and to be sure you have the correct statistical analysis in place. Clear communication with the sponsor regarding the clinical study protocol so that you can ensure you have very high quality PBMCs for the ELISpot assay. Third, to have well-trained staff for the PBMC prep part of the assay, as well as sample processing and the software data analysis portion. And finally, to have ELISpot specific systems in place, such as electronic notebook forms, LIMS, or a validated Excel spreadsheet for data analysis. Thank you so much for your time today, and we look forward to hearing your questions.
All right, well, thank you very much for that insightful presentation. Now I'd like to invite our audience to continue sending in their questions or comments right now using the questions window for the Q&A portion of the webinar. So I've already received a few questions, so I will start with those. So our first question is, I wonder whether you could prepare a dose response curve with ascending concentration of stimulus and then achieving dose-dependent binding results. Diameter around cells may increase. Is this possible?
I don't think the binding results, I don't think you'd yield binding results from doing this. I think that you would be limited by the capacity of the cells. There's antigen-presenting cells within that mix of PBMC. So the real goal is to identify all the antigen-specific T cells that are within that well. You don't want to get too much peptide. It could come out of solution, and you don't want too little peptide because then you're not sure you're getting all the T cells reacting that are in there that are antigen-specific. So I think flow cytometry might be more appropriate if you wanted to look at binding of a peptide.
All right, thank you very much. Our second question, what type of data is delivered to the sponsor?
When we send data out to the sponsor, we include the mean spot number. And if the mean spot number is above the LOD, then we would have a p-value and a response, which is usually a 0 or a 1 coming out of the DFR test. And then any notes as to if there was an anomalous yield or some other special, there might be a note attached as well.
All right, wonderful. Our next question, can you please clarify how the time from blood sample collection to PBMC processing and cryopreservation of PBMCs affect ELISpot assay performance?
Sure. So it's really important. The longer the time between collection and processing, the more granulocytes will be present. So minimizing that time is important to keeping the granulocytes low. Cryopreservation is really the most common way to collect the samples and get them shipped out for ELISpot analysis. So I think, just minimizing that time, and if you can't, use the CPT tubes so that they are processed within 2 hours, and then they can go to the central lab and be either frozen or done fresh.
All right, thank you again. Next question, when would you use CPT tubes as compared to fresh or frozen samples?
I just covered that. So CPT tubes really ensure that the PBMCs are safely handled when you're shipping overnight, and those you want to keep those at room temperature or even a little above, but not cool as you ship with CPT tubes.
All right, thank you. What would be a typical reason to choose for a readout of T cell function other than IFN-gamma?
Yeah, Wendy, I guess you can answer too, but maybe let me kick off there. Yeah, interferon-gamma is very abundant cytokine being produced. So that's one of the reasons why that's often selected. But there may be reasons why you select another one if there is a specific pathway you're interfering with, or for instance, in oncology, you maybe want to evaluate whether your treatment also not just leads to cytokine release, but also effectively has a cytotoxic response. So like you want to assess granzyme B or perforin. I think one of my slides touched upon that to really demonstrate that there is a very effective response. So that might be even stronger. But yeah, I guess it mainly depends on what your drug is doing, what your pathway is that is affected, and how you want to evaluate that best. Wendy, you want to add to that? Or?
No, I think that's perfect. It depends on the question you're asking.
All right, thank you. Are there any examples of application of ELISpot in trials in the field of respiratory medicine? If so, what is the value of using BAL samples versus blood samples?
So that's two questions, and maybe first to touch upon a lung disease or respiratory disease. I think one of the slides I showed was about emphysema and how it's been, ELISpot has been used to explore more diagnostic or pathophysiologic aspect of the disease. It has also been used in tuberculosis diagnosis. I think there's quite some publications on application there. In terms of trials, I know there has been a trial in non-tuberculous mycobacterial lung disease where ELISpot was really used to stimulate the T cells and monitor the response after several days after several months after treatment. And I think, but that's I should need to dive into that. But I remember having seen also a publication about gene therapy for AATD. So again, I think matching with the example I gave, where if you want to monitor the response against the vector, actually, then that ELISpot can be very effective too. So yeah, and sorry, the second question is about bronchoalveolar lavage. Yeah, bronchoalveolar lavage has definitely been applied. And Wendy can maybe allude more to whether how feasible that is or how tedious the sample can be, but it definitely provides more specific information about what happens in the lung, and certainly in some of the diagnostic applications, it makes sense to monitor bronchoalveolar lavage responses using ELISpot to see if someone has been exposed or is infected with a certain pathogen, and there it can be a very sensitive tool to really direct to the spot of infection. Yeah. When do you want to say anything about the sample feasibility, or?
No, we haven't processed bronchial lavage. One thing I can think of is it might be just more dilute. You might have fewer cells, so you'd really need to make sure you are getting enough cells to really see a response in the ELISpot.
Yeah.
All right, thank you very much. Our next question, can the software detect the diameter of secreted cytokine in a dose-dependent manner?
Yes, so the software collects a tremendous amount of information on all the spots. So you can choose, do you want to look at just spot number? Do you want to look at the average diameter, the area? And I have seen papers where they've tried to do a correlation between spot number and diameter.
All right, thank you very much. Our next question, can the precision target criteria applicable for all ELISpot assays, or they may vary depending on the analyte or type of analysis?
I'm sorry, could you repeat the question, please?
Yeah, sure. Are the precision target criteria applicable for all ELISpot assays, or may they vary depending on analyte or type of analysis?
We really developed those based on both the published results as well as the data we saw during method development and qualification. So I'd say it would be, I think they're a good place to start, but you'd want to develop your own based on the performance in your own lab.
All right, thank you. What parameters are usually optimized during method development?
Certainly you can, there are kits on the market and they give you nice ranges, but you want to really check each individual parameter as you go through the different incubations. I also think sample collection and handling stability would be important, especially if you're shipping distances, and to look at those tolerance.
Wonderful. One of our last questions, how many individual donors have to be assessed during method development and validation?
That would really depend what type of ELISpot you're doing and what background. I'm thinking particularly of gene therapy. You want to have enough numbers to really understand what the background is in the population you're interested in. So if you can get your hands on the disease population and then look at the background, say AAV reactivity within that. Recently we did maybe 20. I know the patent paper that Arnaut gave the example, they went up to 100, which is a real investment of time. But it's important to know what your background reactivity is.
All right, well, thank you very much for those answers. We have reached the end of the Q&A portion of the webinar. If we couldn't attend to your questions, the team at Celerion may follow up with you, or if you have any further questions, you can direct them to the email addresses on your screen. Thank you everyone for participating in today's webinar. You'll be receiving a follow-up email from XTalks with access to the recorded archive for this event. And the survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now, please join us in thanking our speakers, Arnaut van Harst and Wendy Adamowicz. We hope you found this webinar informative, and have a great day, everyone.